recording. All right, it looks like it's recording. Perfect. So welcome everyone. Thank you. And I'm sorry again for posting this late, but hopefully it'll get up here tonight. And thanks to the couple of people, to Suzanne and Katie and Floor, who are here tonight watching the live streamed version. So the purpose of this lecture is to give everybody an introduction into the biomolecules that we use to study microbiomes. And I'd just like to acknowledge up front that there's material interspersed in here from a variety of folks, from Kelly Wrighton, Raina Mayer, Matt Sullivan, Kostas Konstantinidis, Ahmed Zayed, and a few other folks noted on specific slides. So this quote, I think, is so pertinent to really all of what we're doing in Emerge, and to the Metaomics portion in particular, which is that education is not the learning of facts, but the training of the mind to think, right? So this is this wonderful quote from a young Einstein. And the idea here is that facts change, our sequencing technologies change, it's like really, really quickly, right? Our methodology changes, the science itself changes. And so, and our need for facts will vary enormously in our lives. But the guiding principles of our inquiry remain useful even as facts wither. And so we can really think about applying that to the biomolecules as an anchor for understanding ecosystems. The specific tools that we use to query those biomolecules will change over the course of our careers. But if we can understand the fundamental caveats involved with different types of biomolecules, which are defined in no small part by the physical nature of those biomolecules, it can really help us think more elegantly and accurately about our experimental design and the nature of the data that we produce. And so if we had time to actually sit in a classroom setting, we would pause for a moment and reflect on some of the facts that might have changed in our own lifetimes in science. Oh, and then this is for the folks watching at home later. I mean, we're all watching at home right now, of course, but for folks who are watching this later, it's really hard to sit and watch videos. I know it is. It's much harder than watching a live Zoom thing. And so I just wanted to remind folks that it's good to pause for a moment and think, hey, what is it that I'm trying to get out of this? Why am I watching this? And then how you can support yourself in staying engaged while you're watching Zoom videos. And maybe that means watching at 1.25x or 1.5x. Maybe that means setting a timer and only watching 20 minutes at a time and then getting up and taking a break. Whatever it is, please try to apply that to this material and the other material that we're providing so that you can really meet the training goals that led you to enroll in the Summer Institute to begin with. Okay, so part one of this lecture is like why we use molecules and how the central dogma kind of anchors that concept. So a central challenge to studying microbiomes is you really can't grow the vast majority of microbes in the lab. The number used to be more than 99%. Now this really varies depending on the ecosystem that you're looking at. We do better and worse in different ecosystems. But even if we look at them under the microscope too, the morphology is not diagnostic. So we see a lot of like little squishy, squiggly things, and that doesn't necessarily tell us what the organism is or certainly what it's doing. And even when we do grow them in the lab, that might tell us something really important about the one particular lineage, but not about what it's doing overall in a community. So this difference between what we can grow just in terms of the number of different types and what we can see by microscopy or indeed by metaomics is called the Great Plate Count anomaly. And this reflects the fact that tame does not equal wild, right? So what you can grow in the zoo of the laboratory is not what we see out there in nature. So instead, we access microbial diversity, metabolic potential, and expressed activity through the lens of molecular biology's central dogma. And this is really the paradigm of sequence-based information transfer in biological systems. This is the replication of DNA, transcription of that information on a gene-by-gene basis into RNA, and then translation of mRNAs into proteins. Okay? So this lens tells us something about identity and metabolic potential through the DNA, and expressed metabolism through the RNA and protein. And those tell us different stories for reasons we'll get into. The things that we actually look at when we're looking at DNA are units of genes and genomes. For RNA, it's transcripts and transcriptomes. And for proteins, it's proteins and proteomes. And so here, what we're talking about when we talk about a genome, we're talking about all the genes for a given organism in its genome. And again, transcriptome would be the entire complement of transcripts for that organism, likewise with proteomes. And we can use this, the words we use for studying these things are gene surveys and genomics and transcriptomics and proteomics. And that just describes the general approach to looking at those things. And so again, DNA is telling us about identity and potential. RNA and protein are telling us about expressed activity. Oops. And when we insert the prefix meta in front of these terms, that denotes community level. So when you hear people talk about community genomics, that's the same thing as metagenomics. Okay. So this is all happening inside the physical space of the cell. So in the early days of metagenomics, people would talk about communities as just a big bag of intermixing genes, but of course that's not really what's happening. Genes are transcribed and translated inside individual cells that have differing levels of genetic connectivity to other cells and populations and communities, and indeed to other organisms in the entire ecosystem. And all of those other organisms and cells have differing degrees of genetic connectivity themselves. So you might have heard of horizontal gene transfer. And so some lineages are really prone to horizontal gene transfer, some not so much. There's different mechanisms for that transfer. One of them is viruses. Viruses, you might think about viruses as agents of mortality, but they also move genes around. Okay. And collectively, it's this suite of interactions from genes all the way up through ecosystems that give rise to system outputs. Okay. And those system outputs are things, you know, for the purposes of our study systems, those are things that we care about, like toxin production in a harmful algal bloom, or greenhouse gas production in our beloved site of Stordal and Meyer, or bioremediation in, say, an acid mine drainage system where we're trying to actually clean things up, right? So the system output could be any range of things. And this is happening in varying time as well as in physical space. And so DNA, the half-life of DNA is hours to years. The half-life of RNA is seconds to minutes. The half-life of protein is sort of on the scale of minutes to days. It should be, these texts should be scooted over to be above these biomolecules. So when we think about organismal response to changing ecosystems, we have to think about how and where they're going to be acting in this information transfer system to mediate that response to change. So one quick example, and I think Ahmed will probably get into this, Ahmed, Sarah, and Mike will probably get into this tomorrow in the regulation section of the agenda, is that it's really cool, actually, when you think about it. If you're a little cell, and this is totally anthropomorphizing cells, and we're not supposed to do that, but it's very helpful for the purpose of conceiving of it, I find. So if you're a little cell and you need to be able to respond to changing environmental conditions quickly, okay, and it's like a highly competitive environment, there's a nutrient that's going to come along and you need to jump on that nutrient before any other organisms do. You might want to have your mRNAs ready to go. You might want to have your whole little flotilla of mRNAs that are encoding that protein ready to go, so that all you have to do is turn on translation. So you might have like a little LoJack system on those RNAs to keep them stable and keep them sticking around a little longer, so that you can go ahead and get proteins really quickly. Or if it's really critical, you might even want to have the proteins themselves around all the time, ready to go. Okay. So this is just to make the point that these different biomolecules integrate across different timeframes. We'll come back to that later. So collectively, these molecules describe who, what, and how of microbiome studies. So I'd just like to flag that there is an optional material slide deck that accompanies this whole lecture. And that's got a bunch of other information that people who want to dive a little bit deeper into this for any reason might find useful. So there's information about why the 16S RNA gene is a good marker, how we use it to get the tree of life that you've probably heard about. What is phylogenetics and how does it differ from taxonomy? How we use single molecules to study microbiomes, which overall is not going to be something we spend much time talking about today, but is a whole field, right? How the 16S RNA gene amplicon studies that are really abundant in the literature get us community profiles, OTUs, and ASVs. We'll talk a little bit about that today, but not much. And then how the molecular details of PCR define the caveats of amplification-based community profiling. And I've tried to stick away from too much methodology today because, as we described earlier, that's changing all the time. But PCR has not changed for a while. PCR is like the workhorse of molecular biology. And so for a whole range of different molecular applications in microbiome studies, PCR is still kind of at the center of those. And so its nuances actually define a lot of the caveats of the data that we work with. Okay. Thinking a little bit about reverse transcription PCR, which is RT-PCR, quantifying things with qPCR and gene probes. So we're not going to talk about those today, but there is another slide deck that I have not walked through verbally, but is designed to be pretty self-explanatory. And for part one, this information is there. Okay, so briefly, jargon decoding. These are terms that you have no doubt heard your microbial ecology colleagues using, and you might have wondered what they meant. So OTU stands for Operational Taxonomic Unit, and that's because species are difficult to pin down in microbes. In macroorganisms, we define species by the ability to interbreed. But you can't really do that for microorganisms that divide asexually, but can, to wildly varying degrees, exchange DNA. So we can't use the ability to exchange DNA to define species in microbes. So instead we have like an operational definition, which is the O in OTU. It's just an operational definition for functional purposes, like species-like categories based on DNA relatedness. For things that you can grow in the lab, you can describe a species based on a shared phenotype that you care about, the ability to infect a person, the ability to transform a particular contaminant, etc. Okay, but in general, species are squishy for microbes. So skipping the historical reasons for why this is true, we usually use 97% identity as like a standard definition for what an OTU is. And that identity means the sequence identity. And here by identity, we mean the similarity, how identical it is. The sequence identity of 16S RNA gene sequences between two organisms. If those are 97% identical or more, we're going to lump them into the same OTU. And more recently, because so much diversity of phenotype in terms of physiology and ecology can be squished together within a 97% OTU, now a lot of these Amplicon studies will use ASV, which is Amplicon sequence variants. And here we're looking at uniquely different, so like 100% distinct sequence variants, okay? So those are some terms that you might hear bandied about that it's good to just define. Okay? So if you've isolated a new bacterium and you get your 16S RNA gene back and it's got 86% similarity to a known organism with a certain taxonomy here, what can you say? So based on that identity, if it were 97% identical, we might call it a species. 95% similar or higher, it might be the same genus, etc. So if it's 86% similar, we might say, hey, it's probably within the same order. Based on our reference sequence database, we might be like, okay, it's probably within the Enterobacteriales. So that's just a quick example. So how does this work in practice? So you've got your full length, this is a schematic of a full length 16S rRNA gene. It's around 1,500 bases. Okay, you've got different conserved and variable regions color coded as colored blocks here. C for conserved, V for variable. The way that this sequencing works is to use PCR to amplify a variable fragment from one of the regions. Which region and how long is defined by the sequencing approach you're using and what other data you're trying to compare it to. When you amplify something, you call the resulting fragment an amplicon because it's been amplified, okay? If you do this on a whole community and then you sequence your pool of amplicons, that results in a mixture then of amplicons that you sequence to produce 10,000 or many, many more amplicon sequences from your community. You then cluster those amplicon sequences into groups based on similarity, either sequences that are identical if you're just doing ASVs or like 97% if you're doing OTUs, and then you identify them by how similar they are to your reference database. Okay, so that's the general principle of this kind of tagging approach. Okay. And many, many human microbiome studies, even now, and the Earth Microbiome Project have been and are done via iTags. And so these are called iTags. And it's I because it's Illumina sequenced. And tags because these are like little barcode tags for the different lineages. Okay, so there's some real caveats about this, and I'm spending a minute here, even though we don't use iTags much within, very much within eMERGE, although we do have them and they're very useful for a lot of things, but there's some really important caveats. And oftentimes you'll see papers published, especially in the human microbiome space, that don't necessarily kind of, you know, that maybe gloss over some of the caveats. So I'd like everyone to be clear on these quickly, just so that, especially as we do work with a mixture in this project of iTag data and metagenomic data, you can really start to become fluid about thinking about the pros and cons of each data type and the caveats of each data type. Okay, so what are some of the caveats with these iTags? So high throughput 16S RNA amplicon sequencing. First of all, PCR is used. So PCR involves primers to amplify specific targets, and those may not amplify everything in your community the same. There may be appreciable biases in the amplification so that you're skewing the abundances of different lineages. Maybe you're missing some altogether, and indeed that does happen. Also, when you have your amplicon pool that gets sequenced, that tells you relative abundances, and again, of the amplicon pool, not the original actual community. So it's relative abundances, not absolute abundances. And why does that matter? Well, microbe A might be 5% of community 1 and twice as much, 10% in community 2, but actually be present in higher cell numbers in community 1 because community 1 has an overall higher cell count. So this is a vital concept, the difference between relative abundance and absolute abundance, which may, to some of you as scientists, you're like, oh yeah, relative versus absolute abundance, totally get it. But when you start looking at this data and you're comparing different samples with your relative abundance, it can be really easy to fall into the trap of conflating relative and absolute abundance. Okay. Another trick about this is when you're looking at 16S tag sequencing, you know, we said it's relative abundance, not absolute abundance. Another layer that kind of influences this too is that the 16S gene varies in copy number. So we said that we're looking at variants of the 16S gene. In a perfect world, the thing that you're using to tag all your different lineages would be present in one copy for every single genome. Instead, oh, I see that there's a chat. You should just interrupt me. If anybody has a question, please interrupt me. Is that an old?
Oh, sorry. I was just commenting on point number 2.
Yes.
The caveats of FTIC-RMS for geochemistry are similar to the caveats of 16S rRNA.
Thank you, Suzanne. That's an excellent point. I think that because this is getting recorded the way it is, people who watch it later won't probably see the chats because I don't think I set it up so they would like scroll on the side or anything. There is some way to do that, but not through here. So it's great. If you have a comment like that, that's a great linkage to another major data type for the project. So please like just interrupt and chat them out. That's very helpful. Okay, so instead of being present at just one copy per genome, the 16S is present, 16S RNA gene is present at multiple copies. On average, it's somewhere like 2.1 copies per genome, but it can range from like 1 copy per genome all the way up to, I think the most is like 24 copies per genome. And so then if you're trying to look at the relative abundance of an organism through the proxy of the abundance of its amplicon sequences of its 16S gene in part of an overall community pool, and one organism has 24 copies of that gene and the other has 1, that's going to definitely change your interpretation. And if you're not clear on this caveat, it could change your interpretation in an incorrect way. You could make incorrect assumptions of what that means. Okay. So, etc. There's other caveats. We don't have time to get into all of them, but those are kind of some of the main ones I want folks to be aware of. Okay, so that's part 1. Part 2. What is metagenomics? What is it not? It's not I tags. And what is metagenomics? Where can we use it and why use it? So reminding ourselves of where we're at with metagenomics, we've got DNA. We can do single gene approaches. That includes the 16S RNA gene amplicon sequencing. That includes I tags. You're not accessing the genome here. You're looking at a single gene, not the whole genome. So it's not metagenomics, it's single gene community profiling. And in contrast, you have whole genome approaches, and this can include community genome sequencing, i.e. metagenomics. And reminding ourselves, oh, this is another caveat here, not really a caveat, but just an inherent nature of the data. So the amplification biases may skew the relative abundances, but then here's the inherent nature of the data part. Function can only be inferred by known relatives. Okay, that's kind of dodgy. So if you have something that's closely related to another organism that does something you care about, you might think, hey, maybe that does the same thing. Maybe it has the same role in the ecosystem. Maybe it performs the same key metabolism that links to my biogeochemistry. For some metabolisms, it does track pretty well with phylogeny. For other metabolisms, it does not. Okay, those would be things that are more likely to be horizontally transferred, for example, or might have evolved independently. Okay, so if you're looking at I tags and you're trying to understand the function of the community, that's got some major limitations. Okay, so how to conceive of a metagenome? It's kind of like you have a lot of different puzzles all mixed together in the same bag. And what's more, there's not just one copy of each puzzle. There's different numbers of copies of different puzzles. Okay, so we're going to come back to that. Why would you use it? Who is there? Okay, so you can answer the who is there by high throughput sequencing of your 16S RNA gene. Okay, and that can get you the tree of life. But it misses lineages that primers miss. You don't see viruses at all because viruses don't carry the 16S RNA gene. Okay, and you can't tell what they're doing. Okay. So now let's contrast single gene approaches and overall genome approaches. So if a single gene approach, if we think about a puzzle of the Golden Gate Bridge, okay, a single gene approach might just look at one individual piece of it. Okay? Whereas a whole genome approach might look at, ideally, the whole genome. You might miss a little bit, but you can look at the whole thing. Now, there is a reason why this is missing. It's the same piece that's missing over here because the 16S gene is underrepresented when you do your assemblies for metagenomes. for a variety of reasons, but that's sort of a technical detail. But anyway, a single gene approach is like one little piece of your puzzle, and your whole genome approach is the whole puzzle-ish. Okay, and so that reflects, in this case, the 16S RNA gene versus a genome. These are not to scale. Okay, how much larger is B than A? So this is about 1,500 bases, and this is about 2.1 million bases. Okay, so 16S does not predict gene content. So just to extend the concept that function does not track 16S. Here's a great example from E. coli. These are 3 different strains of E. coli. So we've got a uropathogenic strain over here in pink. 90% of United States UTIs is caused by this strain. You've got an enterohemorrhagic strain, 0157H7, and this causes blood loss in the intestines, and it can be lethal even in the United States, even with hospital treatment. And then over here in green, you've got this sort of friendly K12 non-pathogenic that's like in all of us, okay? So these are 3 different strains of E. coli. They're still considered the same species by classical definitions of E. coli in clinical settings, but they only share 40% of their genes, okay? But they have near identical 16S. Okay? So this is a great example for thinking about why you can't necessarily predict a particular function that you might care about, in this case, how sick it makes humans, just from the identity of your 16S. Okay, so once you have metagenomes, what do you do with them? You've sequenced all those pieces from a bunch of different puzzles. Now what do you do with them? We're totally skimming over how you sequence them because that changes all the time. So first you need to take those puzzle pieces and you need to start fitting them together. Okay, and then since we have a bag of lots and lots of different puzzles that are mixed together, we don't just want to like put the puzzle pieces that actually fit together. We want to try to figure out, oh, look, all these pieces go from the same puzzle. These pieces go from a different puzzle. Then we want to predict the genes. Okay, now we start to move away from the puzzle analogy. We want to actually predict the genes. We want to annotate those genes. So once we've identified where some of those genes are, like what might they be encoding, okay? And then predicting the metabolisms from those genes, the metabolic pathways, and indeed the metabolisms for the overall organisms, okay? And then we can quantify the organisms and the metabolisms by mapping our metagenomic reads back to these assemblies, okay? And then we can start doing things like comparing relative abundances, which is how we get to ecology. You have to be able to count a thing and look at its differential presence and absence and abundance to be able to do ecology. Okay, so first assembling those raw reads, fitting the puzzle pieces together. Okay, so you can match the pieces up. So we're going from this, just a mixture of puzzles. Remember, there's many, many copies of this. So the right way to think about this, oh, those came out as black. They shouldn't be black. The right way to think about this is it's more like there's many, many different puzzle pieces kind of all layered on top of here, as well as connecting like this. You're like, okay, I can see that this is how this all fits together. Okay. This one is pretty complete. This one is less complete. And then we've got random pieces of other things around here. We do notice here that like, oh, look, maybe those are from the same puzzle, but we haven't really gotten to that point yet. Here, we're just looking at contigs, so things that are actually physically connected. So contig stands for contiguous DNA. Okay, so one of the challenges with this step that kind of defines some of the things we think about with metagenomes is that the abundance per genome varies a lot. So how many copies of each puzzle are in there? And let's skip this in the interest of time. So if we just look back at this, and if you had to, like just this part of it over here, if you had to guess which puzzle you have the most copies of, it's so hard to teach to pretend you guys aren't there and like not ask you questions, but you're all juggling. The three of you who are here are juggling things. So I'm not going to try to ask you questions, but everybody watching at home later, think which of these puzzles might be most abundant and why would you think that? Okay. Okay, so then after assembling the reads, you've got something like this. Oh, here we go. Yeah, which organism would you hypothesize to be the most dominant or the most rare? I.e., which puzzle is there the most copies of versus the fewest copies of? But what assumptions do you have to make in order to make that connection? Okay. And that brings us to another major assembly challenge. It's not just that the copies of each puzzle vary, it's that the size of each puzzle varies. Is it a 50-piece puzzle that we're doing with our little kids, or is it a 5,000-piece puzzle that we're doing with our whole family when we get together for a weekend, right? So genome size varies a lot. So here's an example of that on the x-axis. We go from higher eukaryotes all the way down to viroids, things that aren't even actually viruses, through different kinds of viruses, through bacteria, quote unquote, lower eukaryotes and higher eukaryotes. Okay. And I left this, this axis in here because this is no longer true. And so anybody else who, like me, thought it was true because you learned it was true at some point, I just want to tell you it's not true anymore. The idea that viruses have faster mutation rates and like bacteria and eukaryotes have slower mutation rates is not actually true with the current, more robust data. Okay, so if you're trying to assemble all these different things together, and some puzzles are little teeny tiny and some puzzles are really big, that challenges your ability to compare among the different puzzles in a consistent way. The other thing is microdiversity across related genomes varies among lineages a lot, and smart people are working on solutions to this, okay? So a way to think about this with our puzzle analogy is, are the copies, the different copies of the puzzle, remember we said this is a big mixture, a big bag with lots and lots of puzzles in it. There's many copies of each puzzle, but are those copies identical, or are they just similar, or are they a little different but could maybe still cross and form how you put them together? Okay, so a way to think about this is like over here, you've got 3 puzzles of the Golden Gate Bridge that look pretty similar. Okay, they're in color, the bridge is from the same kind of orientation, the bridge still looks pretty much like the bridge in all of them. Okay, now over here, you have higher microdiversity, let's say, right? So you have different kind of different looking bridges here. They do all happen to be the Golden Gate, and there's some iconic similarities here that you can relate to each other, okay? But they're not near as similar as on this side. Okay, so now if we look back at the contigs that we've put together, and we go back to our question that we posed before about which organism would you hypothesize to be the most dominant or the most rare, you might have hypothesized, well, the Golden Gate Bridge puzzle is the most dominant, okay? But now we can think of a couple of other explanations that might explain why the Golden Gate puzzle is the best contig here, okay? Is the best assembled puzzle, okay? So maybe it has a small genome, right? So maybe it was just really easy to put together. Maybe there was only like 10 pieces, okay? Maybe it has low microdiversity. Maybe all the copies of the Golden Gate puzzle really looked identical. So anytime you had multiple copies, they could kind of fill in each other. Okay. So the next step after assembly is binning. And this is where we take our puzzles and we try to put them together, even if they don't touch perfectly. So coming back to our example over here, the contigs were contiguous pieces of DNA, but some of these contigs, that piece, and that piece came from the same puzzle. These were all from a puzzle of the Eiffel Tower. Okay, so there was different features about it, maybe some color, maybe some shape. In the example of the puzzle, we use other things in metagenomes, but are there features that can allow us to put these contigs into the same bin? Okay, even when there's gaps. Okay. Okay, so one of the ways that we do this, and also a way that we come back to later for doing ecology, for just understanding how abundant these different organisms are, is we map our original metagenomic reads to our contigs, or it could be to reference genomes based on sequence identity. So now we go back to all of the sequencing reads that we've got, and we map those on. And by mapping, we're trying to match them up based on identity, based on sequence identity, to a contig or a reference genome. So if we think about our puzzles, let's say we've taken a photo of our completed puzzle, okay? And now we go back into our mixture. Let's say we mix them all up again. And now we take our photo and we stack all the puzzle pieces that match that Golden Gate Bridge photo that we're like, okay, we assembled that. That was a good genome right there. That nice Golden Gate Bridge genome. And now we're putting all of the pieces that went with that on top of there. We did initially use them to make that, but we weren't kind of counting as we went. We were just trying to make the best Golden Gate puzzle assembly and then bin that we possibly could. Now we want to know, what are all the pieces that match? Maybe some of those pieces we never even used for the assembly and the binning because like they didn't pass some threshold. But now that we've got the reference there of like the full thing, we can see, oh yeah, that actually is part of the Golden Gate Bridge puzzle. And so we want to match those up to where they go based on some criteria of how similar it is to the Golden Gate, okay? And then we want to count them, okay? We want to count how many there are in a given area and overall, okay? And so the pieces here would represent the pieces of the puzzle. And so then we can also assess coverage. So we're talking about horizontal coverage. Are all the parts of the genome covered? Do we have like one of the towers of the Golden Gate Bridge covered, but not the whole rest of it? Maybe that would indicate there's some kind of issue there. And also the depth of coverage. So this is an ambiguous term that can be used, and this is one of those weird jargony terms that can be used lots of different ways in different disciplines and different contexts, even within disciplines. So the depth of coverage here means how many pieces on average map to any given part of that Golden Gate puzzle, okay? And then horizontal coverage is like how much of the puzzle overall is covered. So what should the coverage depth be proportional to? The abundance of that organism. Okay, so what should the copies of the puzzle pieces be proportional to? It should be proportional to how many copies of that puzzle were in the mix in the first place, which should track to getting back to our actual community and the organisms, how abundant that organism was. Okay, so abundance, all else being equal. Okay, so then we can actually use abundance to bin contigs together. Okay, I'm not actually going to talk about that right now because I'm just going to say that that is a thing in the interest of time. Okay, and then when we have a bin, a genome bin, like that Eiffel Tower thing where we've got different contigs that we've identified all go to the same puzzle, how do we assess how complete and how pure that genome bin is? The way to do it is to assess single copy marker genes. So here, the columns are different genome bins. So each of these columns represents, like this column could be the Eiffel Tower, this column, where's my arrow? This column could be the Golden Gate Bridge, except it's not because there's too many gaps, and this column could be the Colosseum or whatever, okay? So that's just an example, not really specific to those columns, of course. The rows are different single copy marker genes. There's something like 120 single copy marker genes that are robust for bacteria, okay? And then you can say, hey, is that single copy marker gene, and by which we mean a gene that is present only once in a bacterial genome. They exist. It just happens that 16S is not one of them. Like the field started out doing 16S for a lot of good historical reasons. It's just not a great marker gene for other reasons. It's not single copy, but there are other single copy marker genes. Okay, so that's what we're assessing here. In this visual representation, the box color of each of these cells from the rows and lineage, or the rows and columns, if it's green, that means it's present. If it's gray, that means it's missing, or red means it's duplicated. You should not have 2 copies of a known single copy marker gene in your genome bin. If you have 2 copies, that's telling you that your genome bin is contaminated. Part of this Colosseum puzzle snuck into your Eiffel Tower puzzle. It should not be there, okay? Okay, so red means you've got some contamination. Gray means you've got some single copy marker genes missing. Okay, so we can use this approach to tell us about completeness. So, and also redundancy, as we just described. This does not work for viruses because we don't have a set of single copy marker genes for viruses. Okay, normally I would be stopping for questions, but since our audience here is small tonight, please interrupt me if you have questions. So now to get to the key piece of jargon that you hear us talk about all the time, a genome bin that is good quality, we call that a metagenome-assembled genome. It's conferred the status of being called a MAG, a metagenome-assembled genome. Okay, the exact cutoff for that varies based on the paper, but we could say that, for example, it might be at least 70% complete. So at least 70% of those single copy marker genes are present, and less than, say, 5 or 10% contaminated. So less than 5 or 10% of those cells would be red, that you have more than 1 copy of a single copy marker gene. Okay, so now we've assembled and we've binned. Now we are going to predict genes and annotate. Okay, so predicting genes. So here we're predicting our open reading frames. An open reading frame refers to the part of your DNA that gets opened up for replication and is, actually, no, that's not that part. This should be over here. That is opened up during transcription and is actually read into mRNA. So that little arrow, that little line should be moved over here, okay? And so that open reading frame is the piece of DNA that corresponds to one particular sort of piece of instructions for the cell. And we can recognize those by specific sequences that say, hey, this is the start of a gene, and hey, this is the end of a gene. Okay, so then you can take your open reading frames that you've annotated in your MAG, your Metagenome-Assembled Genome, and you can say, what do they do? And here you use a suite of tools based on the similarity of the primary sequence. Okay, and here we usually look at the amino acid sequence. So we take our DNA and we predict what it would be in protein based on the translation table for how the DNA gets translated into proteins. We know what that translation table is for biology. So we can say, hey, here's our DNA sequence. Here's what the protein would look like. And then we try to relate that protein to a bunch of other known proteins that we know something about. And that's how we make functional assignments. And we use a whole different range of tools because different tools have different strengths and weaknesses. So it's based on the similarity of the primary sequence of the actual amino acids in the protein, as well as the secondary and tertiary structures of how we would predict that protein folds up. Okay, we also looked at things like conserved motifs, like, oh, okay, this is the active site of the protein. So this is super highly conserved among all the different variants of the proteins that are out there for that particular protein type. So the active site might really be conserved, and then the rest of it might wiggle around a lot more and be less conserved. So we can incorporate that kind of information into our searches and into our into how we functionally assign function to those proteins. Okay, so some rough rules of thumb. We're going to skip that for now. It's there if you want to look at it. A quick thing to remember, though, is a single amino acid change can change function. Okay, so that is a huge caveat with assigning function. Okay, so now you've got your ORFs, your open reading frames, and you've got predicted function for all those genes. Okay, and now you can start to infer the cell's potential metabolic map. So you have like a template from E. coli, for example. And you might say, well, how similar is my MAG to all the different metabolic pathways that we see in E. coli? It will have some that E. coli doesn't have that you then have to sort of map on, and it will have some that E. coli does have, and it will be missing some that E. coli has. But you can think about it sort of like a rough scaffold to start throwing, there's an expression about throwing spaghetti at the wall and seeing what sticks. So you can think about, you've got all of your genes and you're trying to throw them into an overall portrait of how the cell's metabolism is working. Okay, so once you've gone through that, you can actually start to do ecology. And there's different levels of ecology that we do. Okay, we can talk about doing gene ecology, lineage ecology, genome ecology. So let's describe what those mean. So gene ecology is looking at how the genes and functional groups of genes, like all the genes in the antibiotic resistance category, are differentially distributed among your samples and how their abundances relate to habitat characteristics. We can look at lineage and guild ecology. Okay, and here we would be doing the same thing, but for specific lineages like Methenoflorins, Stordella myrensis, our favorite lineage from the mire, or functional groups like all methanogens. Okay. And then we can do genome ecology. So now we might sort of zero in to a specific MAG. So now we might look at, we were talking about Methenoflorins before. So Methenoflorins, Stordella myrensis might be a species. But we might look at one particular MAG within that species and try to understand its ecology, okay? And it's by doing things like that that you can see, hey, there's actually different strains here because different MAGs that should be kind of the same species actually have different ecological distribution. That suggests there's something going on with strain variation, okay? You can also, again, group those MAGs back into functional guilds. Okay, so what information do you need to do ecology? You need to define what you're tracking. Are you tracking a gene? Are you tracking an organism? Are you tracking a functional guild? And you need to be able to count the relative abundance of that group. How many trees of kind X are there in the forest? How many copies of organism X are there in my microbiome? Okay, another key thing you need to do ecology is you need to say, hey, is my forest a dry forest? Is it an alpine forest? You need sample metadata. So things like pH, oxygen, host disease state, location, all those things would count as metadata. Okay, so rhizosphere versus bulk soil, etc. So there's a related slide deck. The optional slide deck has information related to part 2 that has a little bit more about strain variation, a little bit more on binning, some ITAG versus MetaG compare and contrast, and then a bunch of common MetaG analyses. Okay, so Venn diagrams, heat maps, ordination, network analyses, a little bit of information about statistical linkages to metadata. And this is going to be briefly covered by Matt this week as well, and some information about new paradigms from metagenomics. Okay, so you find unexpected things almost by definition when you look at the entire community and you don't filter your approach for the things you're already interested in. So funny vignette about this, there's kind of this, whoa, what did I just do? That was weird. Okay. Funny vignette, like there's this cultural difference that people from a metagenomics background are like, hypotheses. Like if you've got a cool experimental design about looking across different metadata gradients, like you're gonna see something cool, right? And then if you come in from like the NIH world or a lot of different parts of biology, it's like you have to have your hypotheses really, really clearly articulated. And so there can be this difference in how we write proposals. There can be this difference in how we write papers. And in the early days of metagenomics, it was very exploratory. And there was a lot of sort of challenges with that cultural disconnect. It's like, hey, if you're exploring other planets, you're going to find amazing things, right? So, so one emergent finding from metagenomics is really that it's not just about centrophys. We heard a little bit about centrophys today from Jeff in one of our lectures. And it's not just about pairwise centrophys. Really, metabolism in communities is like an entire web. So there's a ton of sort of centrophic interactions going on. Centrophy kind of implies this just one-to-one. This is more like intricate cross-feeding. Okay, so microbes in the sea, for example, are really like cells in a body. So there's all of these different cells and they're in a little, milliliter of seawater or whatever. And so these metabolisms are often studied one cell at a time. Okay, but really the microbial communities as a whole can form distributed metabolic pathways. And you can compare those between environments and say, hey, this part of a metabolic pathway for the overall community seems to be missing in this environment versus this environment. And a key linkage here then is this kind of community level metabolic inference tells us something about the biogeochemistry that might be dominating in environment 1 versus environment 2. For example, if you don't see methanogenesis represented in the overall community metabolism for an environment, it's a pretty safe bet you're not going to be getting methane from that environment. Okay, and if you see methane coming out from that environment, then either there's another mechanism that's not been described before genetically, which happens, like that's happened recently that we've seen new ways, okay? Or maybe the methane is coming from a different community below or to the side of the one that you're studying. Okay. And so we'll skip this. You can look at this at home if you want. Right, so this idea that connecting metagenomes to distributed pathways to biogeochemical cycles is a current frontier in our field. Okay, last section, part 3, introduction to metatranscriptomics and metaproteomics, and what are some of the technical and analytical considerations of each tool and what you could learn from each. And then there's some optional slides, which is again a separate slide deck. Ahmed's regulation summary figure from the proposal, a tiny bit of history about metatranscriptomics, how methodologically you do RNA removal, some more discoveries from MetaT, and technical details of MetaP. And we abbreviate these oftentimes as MetaG for metagenomics, MetaT for metatranscriptomics, and MetaP for metaproteomics. Okay, so we've covered who is there. So we take a microbiome, like a gut microbiome, we sequence the amplicons of the community DNA, and we categorize who is there. We map the predicted functions based on gene content into metabolic webs. Okay, and then we think about what could they be doing. You can't just tell that from the 16S gene, but with the metagenomes overall, you can say what could they be doing, but that doesn't tell you what they are doing. So now we would like to sequence the RNA, characterize the proteins to tell us something about what they are doing, okay? And that's what they're actively doing under current and different experimental conditions. And a quick reminder that there are other tools. It's not just like the metaomic world, and it's not just MetaT, MetaP. So you could look at microscopy, right? Oh, look, this particular organism lives inside epithelial cells. Well, that starts telling you right away something about what they might be doing. But if you just did a metagenome, you would not, like that's a key piece of information, right? That a metagenome would not tell you. Okay, sorry, let me go back. And this is a great example. People, this group Jessica Mark Welch and colleagues a couple of years back generated this 3D map of the gut microbiome, and they saw that it was not discretely layered communities. It was like this incompletely mixed bioreactor. So this is just a flag that tools like microscopy are really helpful at these questions of what are organisms doing. We're not talking about those today though. Okay, so for expressed activity, we're talking about meta-T and meta-P, reminding us of how this fits on this framework. And I'd just like to flag here, metabolomics is also really important. We are not talking about that in today's lecture, but metabolomics is which metabolites, both the intermediates and the products, are present, and how do they vary ecologically. And those are kind of the output, if you will, of the action of the proteins on the environment. Okay, so reminding ourselves that mRNA and protein have different half-lives, and so they integrate over different timeframes. So RNA tells us something about quick timeframes, and protein is a slower timeframe. Okay, there's a whole suite of challenges associated with metatranscriptomics. We're gonna skip those here because of time. I will briefly pause here and say that mRNA is a teeny portion of the biomolecules in the cell. Okay, so it's 0.5 to 5% of the total RNA pool in the cell, and the RNA is only a small portion of the total biomolecules in the cell. So if you want to go after the mRNA, you have to be methodologically clever. Fortunately, there's a lot of clever methodological folks out there who figured out good ways to deal with this. Okay, and the methods for those are detailed more in the optional slides. So just to go back here, you end up, if we're thinking about our puzzle analogy, you kind of end up again with a lot of different pieces of the puzzle. Okay, so you've got a lot of different pieces. What do you do with those pieces that are your metatranscriptome? And not just one puzzle, but a lot of different puzzles mixed together. And let's say, if we're thinking about the puzzle analogy and we're trying to apply it to metatranscriptomics, and we said that mRNA is just a fraction of what you might be sequencing, for example, in this case, let's imagine we've got some puzzle boxes and stuff in there. We don't want the boxes, but those puzzle boxes got in there, and maybe half of our signal is puzzle boxes, and we don't want the puzzle boxes. We just want, in this case, the metatranscriptome puzzles. Okay, so, but we can find those puzzle pieces, we can take them away from the boxes, and now what do? Well, we QC the sequencing reads. I didn't even mention this for the metags, but we do that too for those. We get rid of the puzzle boxes, so we remove the residual non-coding RNA sequences. And now we're going to go through that same mapping process of looking at how those pieces map on to our reference genes or genomes or contigs or mags. Okay? And here we're looking at the transcriptomices. So let's say, getting back to our good old Golden Gate Bridge puzzle analogy, maybe it's all about those big central towers of the Golden Gate Bridge. And so there is so much transcription going on there. All the pieces are going to those central support towers. Very cool. That tells us, hey, those towers are being highly expressed because so many of my pieces in my metatranscriptomes are just piling up all around those towers. You need to have your reference that you're relating these transcripts to, these puzzle pieces to be able to say something about, you know, what they're like, which organism is expressing them, which gene is being expressed. You can also directly assemble from your meta-Ts. We won't talk more about that now, just it is a thing you can do. A small caveat here, because genes have different lengths and you might have sequenced more deeply in one meta-T versus another, you need to normalize for those things in your output data. Okay, so metatranscriptomics can tell you things like who is most active. let's ignore the particular details here. What we've seen in Stordal and Meyer and a lot of other sites is that the most abundant lineages are often not the most active ones, and the most active ones are not the most abundant ones reciprocally. Okay, getting back to this integrating across different timescales point. Okay, this is a really key idea. So DNA relative abundances. This is actually like what I'd like to do is like make better slides just for this part and then spend a little longer on it. But this has sort of slowly been crystallizing over the last couple of years when I talk about this kind of material. And it's really important. And I'm excited to help discuss it together. Okay, except that we're not really discussing things. But imagine that we're having a discussion. So your DNA relative abundances in your ITAG data or your metagenome data reflect a combination of influences, okay? So these are cells that grew rapidly and really are active now, okay? But your relative abundances in your DNA from your community also reflect cells that grew rapidly a little longer ago, and they're not active anymore, but other lineages' abundances haven't overtaken them. It also includes cells that grew a longer time ago still, but are really stable. So things like spores. An extension of that might be cells that died less than other cells in the community in some recent die-off event. Let's say there was a horrible osmotic shock event, or antibiotic treatment, or a phage attack, and a lot of other things died, and these cells just didn't die for some reason. So maybe they weren't even being active, they just didn't die. It would also include DNA from cells that are dead, not even spores, not like resting states, but are entirely dead, but preserved really well, which might be a particular concern in, say, peatlands. Your metagenome might also include extracellular DNA, not from currently alive cells and not even from dead cells, but just DNA that's floating around that hasn't been chopped up yet, which is a thing. So for these last two, cells that are dead but preserved and extracellular DNA that has been preserved, habitat properties play a big role. So peatlands preserve things like the bog bodies. If you haven't ever seen bog man, aka Tolland man, or other bog bodies, Google it. If you're new to these kinds of environments, it's really interesting. And if you think about the preservation of those kinds of bodies, you can imagine too, like, oh yeah, DNA and little microbial cells probably preserve too. Okay, clay-rich systems preserve a lot of DNA, etc. So those are a bunch of the considerations for the range of timeframes that are all integrated together that give rise to the community profiles that you see in your DNA profiles. What about your transcripts? Okay, these are your mRNA relative abundances. That's what we're looking at in our metatranscriptomes. And these reflect a current response. So this may be dynamic. So this may be induced by the sensing of some environmental change that turns on some gene expression. It's like, oh, hey, I now see this nutrient is here, cool. I'm going to transcribe and translate the gene that helps me process that nutrient faster, again, to anthropomorphize cells. Or it might be some gene that's turned on all the time, which is called constitutive expression. So it's all the time on. Now that can be true for some with their quote unquote housekeeping genes, because like they're doing the housekeeping of the cell, things that always need to happen in the cell. Okay, or it can be other genes that we wouldn't call housekeeping genes, but are still turned on all the time, constitutively expressed for other reasons. Okay. Okay. And a side caveat here, sometimes you'll see people, I'm not going to talk about that. It's a thing. If you're interested in those kinds of details, you can read that. Okay, now the protein relative abundances reflect current and recent response. Again, this can be dynamic or it can represent constitutive expression. And the half-life of the proteins is longer than the half-life of the mRNA. Okay, so they both have that either dynamic or constitutive caveat. Both those things could be the case. And one is integrating across a longer period and one is integrating across a shorter period. And then DNA is like a longer period. Okay, we're going to skip that discovery story. It's cool. It involves our own Jean Tyson. And I remember looking over the shoulder of my lab mate when she was looking at this and we were like, huh. And so without telling you anything more about this, I would just like to say that I heard this great quote and recently, and it made me think of this, that one of the most exciting moments in science is not when someone goes, Eureka, but when they look together with their friend and they go, huh. Because like, that's when some really exciting discoveries happen, when you look, you're like, huh, what does that mean? That was this paper. Which ended up being a Nature paper. Yeah. So that was a major biological advance. It's cool, and it's from Metatranscriptomics. Metaproteomics, we're in the home stretch here, guys. Metaproteomics, this uses liquid chromatography, mass spec, tandem mass spec. So you take your microbial community, you extract all the proteins, you have your mixture of proteins, you digest them, so you cut them up into pieces, okay? Now you've got peptides. Peptides are just little pieces of proteins. Now you do liquid chromatography to separate them, and then you do electrospray ionization to put them into your mass spec. And then you isolate your ions of your peptides, you collide your ions to dissociate, you collect your fragments in a tandem mass spec, and then you get spectra. Okay. Now, at several points in this process, you have the potential for biases. Okay, so in terms of what proteins you can extract, in terms, just to highlight a couple, in terms of which peptides actually ionize well. Those are just a couple of places. Now you get your mass spectra out the end, okay, and then you have to match those to a reference database. Okay, so we can't see all of the proteins. Why can't we see them? Because we lose different pieces or different portions of our metaproteome at different stages here. So when we think about how comprehensive a metatranscriptome is, it's pretty darn comprehensive. Okay, there's far fewer winnowing away points with a metatranscriptome than there are with a metaproteome, where you would like categorically miss things. Okay, so how does this actually work? Here's some pictures from an acid mine drainage environmental sample. So you've got your proteins, you digest them to peptides, you run them through your LC-MS, and you get your spectra. Now what? Well, in parallel, you've extracted your DNA, you've sequenced your DNA, you've done your gene calling and your protein prediction from your genes. And so you have your predicted protein sequence. Now, in silico, in the computer, you take your predicted protein sequence from the DNA, from the metagenomes from your community, and you imagine if you had your trypsin and you cleaved it all up into your predicted peptides, you'd be like, okay, well, what would the spectra of those predicted peptides be? And you match your observed spectra to your predicted spectra for this particular community. And that's how you can identify the proteins that are there. And this is another reason that there's that what we observe robustly in our metaproteomes is much kind of spottier than our metatranscriptomes, for example, because there's a lot of kind of many a slip twixt cup and lip. There's many a stumbling block you can make here. Okay, like there's post-translational modifications, and in general, for example, and in general, those are not considered when you're relating your spectra to your peptides. Sometimes they are, but it takes more computational power. And there's a lot of different variants you could have. And so most people don't do that. Okay, so to summarize the questions that each of these methods answers, I'm not going to recap this. We will just end with looking at a key figure from Woodcroft and Singleton et al. 2018. So Everybody, I think, has read this paper. This is one of the assigned pre-reading papers. Let's take what we've learned from today's lecture and apply it to decoding a piece of this crazy, beautiful figure that the team put together. Okay, so first of all, let's just look at these bubbles. Okay, so each of these little mini bubbles represents a different mag, so a different metagenome-assembled genome. The size of that little teeny bubble reflects the relative abundance, which we now know from today's lecture. That relative abundance was assessed by recruiting reads from the metagenomes to that mag. Now, with the brown outline, this represents pulsa, green, that represents bog, and blue represents fen. Okay, so that given mag there had this relative abundance in the pulses overall. And I think this is like average across all of the pulsa samples, okay? So if you recruited the reads from each separate sample to that mag, and then you took the average relative abundance was at 0.01% of the community, 0.1% of the, and let's be precise, not community, 0.1% of the reads, 0.5% of the reads, etc. That defines the size of that little mini bubble, okay? This bubble over here has the same color, okay, as this one. Look, we're looking, okay? It's a smaller bubble, so its relative abundance based on read recruitment is smaller. It's the same color because it belongs to the same phylum. Okay, so based on the genes that were present in that mag, we were able to, Ben Woodcroft and colleagues in Genes Lab were able to assign that mag to the phylum Acidobacteria. Okay, so that tells us something about the different bubbles, their size, their colors. You can see that there's different mags from different phyla over here, okay? And they just showed, like, I don't know, there was some cutoff, probably 0.01%, but I think it's actually not that. There's some cutoff based on which mags are actually represented here, okay? And here, these mags shown in this portion here are mags that have the potential to degrade xylan. So now again, from today's lecture, we know how the team did this, okay? So they took the mag and they called the genes and they annotated the genes, and then they looked at the genes involved specifically in xylan degradation. And for every single mag, they said, does it encode xylan degradation? Check or minus, okay? If it did encode xylan degradation, it goes in this section over here. So if a genome is in this section, does that mean it can't be in this section over here for cellulose degradation? No, it could be in both places. A genome could encode both xylan degradation and cellulose degradation. And you could make some different figure that represents that. This figure does not represent that, okay? Okay, so that starts to give you a sense for what this figure shows. And then you can start to do things like oh, look, the colors get more like evenly spread out as you go to the fen. And that reflects the fact that richness and evenness both go up in the fen for the microbial community overall, and within these particular functional guilds of xylan degraders and cellulose degraders and xylose degraders, etc. Okay, so this is a nice representation. The reviewers came back and said, aha, but are they actually doing these things? And so initially, I think that none of the expression data or just the metatranscriptome data had gone into this. Once the reviewers came back, the metaproteome data got folded in as well, thanks to Ahmed. And so Ahmed worked with Joel to combine both the metaproteome and metatranscriptome data into these graphics. So now here, the thickness of this line represents how abundant that metabolism was in the metatranscriptome. Okay, how much did you see xylan degradation And here we could do this for every single different mag. And indeed they did. To make this figure, they did do it for every single different mag. Okay, but to represent it on the figure in the end, they took the tally for all of the different mags and they overall averaged it. Okay, so is xylan degradation being expressed in the pulsa? Is it being expressed in the bog? And is it being expressed in the fen as connected back to all of these mags identified here? And so the thickness of the line reflects the transcript abundance normalized. Remember, we said it had to be normalized in different ways. Okay, so it's normalized. And then because of those caveats, remember we talked about like, oh, with metaproteomics, there's a lot of things you can miss. It's, you know, so here we decided to do like detected versus non-detective instead of making it quantitative. And so here it's just like dashed line versus solid line. Okay, so that's what that means here. Okay, so dashed line means it was not detected in the metaproteome. Solid line means it was. Okay, so in conclusion, yay, biomolecules. They're really powerful and they can tell us lots of things. There are many caveats. You have to know what your data is telling you and what it's not. But it's really an elegant lens into how these communities are manifesting life and interacting with each other. And then we didn't cover metabolomics and lots of other very useful tools for G2E, which is genes to ecosystems, that side of things. All right, there we go. We're still recording. Does anybody have any questions briefly while we're recording? Otherwise, I will stop recording. All right, so to folks viewing this later at home, thank you so much. I know that was an hour and just about 20 minutes of intensive biomolecule description. We went really fast, but hopefully it was a useful overview. So thanks so much.
Thank you so much.